Paternal benzoapyrene exposure affects gene expression in early developing mouse embryo. The offspring's health relies on genetic structure of the parental germ cells, as the paternal genome appears to have a key role. For example, in de novo mutation in some of the genes may have come from the paternal side, whereas epigenetic modification of DNA and histones are recurrent in the paternal gonads. In today's environment, there are many contaminants that affect the integrity of germ cell distribution, such as the polycyclic aromatic hydrocarbon PAH, known as the benzoapyrene BAP. It is believed that transmissible effects may arise from accumulated DNA damage in mouse germ cells, which beyond a critical stage of spermatogenesis will have a limited capacity of DNA repairs. Problem with BAP is that it has carcinogenic and mutagenic potential, which also has an impact in the reproduction. To test the theory of how it affects the reproduction process in its offspring, an experiment had been carried out using the in vitro fertilization, IVF. In this IVF experiment, male mice were exposed to an acute dose of the BAP, and using their sperm cells, the scientists were able to study and understand the effects of the paternal BAP exposure on early gene expression in the developing mouse embryo. The gene expression in the fertilized zygotes and embryos was detected by reverse transcription and the blastocyst stages of embryo development. The findings show that paternal BAP changed the expression of numerous genes in the developing embryo, especially at the blastocyst stage, so that some genes were affected at an earlier stage. Enzymes convert the BAP into reactive metabolites, which then covalently bind to the DNA. Exposure of the BAP may then affect a number of cellular processes.
Zygotes and early embryos are taken from 5 pre-implantation stages. Pre-implantation is when the embryo has not yet implanted in the wall of the uterus. The zygotes and embryos are then fertilized either with sperm from BAP-treated male or from control mice. These were used to study the effect of paternal exposure to BAP. The results were then analyzed using a variety of data. One of the following data are called hierarchical clustering analysis. This analysis is based on similarities in gene expression. Samples are horizontally labeled based on the developmental stage they belong to during the gene expression. Vertically labeled genes indicates more than twofold significantly downregulated expression levels at the blastocyst stage. By visual inspection of the clustering dendrogram, we observed that blastocyst stage samples clustered close to each other, and most of the genes at this stage were downregulated of the following paternal BAP exposure. The second data is the functional classification of the genes, which shows a graph of relative gene expression at various developmental stages for A. The graph A shows genes involved in DNA damage repair. B shows genes involved in cell cycle regulation. C shows genes involved in embryo developmental, and the last graph shows that genes are involved in methylation and acetylation pathways. Expression in embryos of exposed fathers is shown relative to expression in control embryos. There is a tendency towards an upregulation of many genes at the 8th cell stage. Still, we found no sign of statistically significant pathway-based enrichment in the effect of BAP exposure. The last data showed differentially expressed genes which are between the control embryos and embryos of exposed fathers at various stages of embryo development.
Zygotes and early embryos from 5 pre-implantation stages, fertilized with sperm from either control or BAP-treated male mice, were used to study the effect of paternal exposure to BAP on the expression of a panel of 78 mouse mRNAs. The aim of this study was to analyze gene expression patterns and to explore the early embryonic transcriptional effects induced by paternal exposure to environmental chemicals. Results indicated that the 8-cell stage may represent a sensitive stage of mouse embryo development for chemically induced changes causing upregulated gene expression. For example, about 88% of the genes were upregulated in embryos derived from BAP-exposed fathers, compared with control at the 8-cell stage. The clustering of 41% of the embryos exhibited significantly upregulated gene expression, whereas others expressed a more balanced pattern, as seen in Figure 1. These cluster samples could reflect embryos with more severe lesions, as the BAP-exposed sperm is assumed to inflict variable levels of damage on the cellular machinery in developing embryos. One gene in particular which is upregulated at the 8-cell stage is PDRG1, as seen in Table 1. The expression of this gene has been found to be upregulated in multiple malignancies, as it is a novel cancer biomarker. 4 different BAP dysregulated miRNAs were found to target PDRG1. The experiment also indicates increased activity of DNA mismatch repair due to the upregulation of the protein PMS1. The experiment also showed a largely uniform downregulation of genes at the blastocyst stage, as seen in Figure 2, although there is not sufficient evidence to suggest that this is due to exposure of BAP. DNA demethylation during the first cell divisions after fertilization of both the maternal and paternal genome is vital. Any interference with this process may affect embryo development or offspring phenotype. Exposure to BAP showed significantly altered gene expression levels of those related to DNA methylation, which may interfere with the epigenetic status in the early embryo. The effects shown in this experiment, although conducted in a narrow timeframe between exposure to BAP and sperm collection, are conducted by sperm residing in the epididymis. Based on the data, it is unclear as to whether the effects of BAP are due to adults in the sperm DNA or if they are the result of other sperm modifications. This is because spermatogenesis is susceptible to change by contamination of chemicals. Although the altered gene expression could be induced by the presence of DNA adults in the sperm DNA or epigenetic modifications, this experiment demonstrates that paternal BAP exposure of epididymal sperm significantly affects subsequent gene expression in the developing mouse embryo. In considering the vast number of changes which occurred in gene expression, this study concludes that paternal exposure to BAP leads to altered gene expression in pre-implantation mouse embryos.